Okay, hello children, am I? Yes, finally. So if at all I say sorry, will you forgive me by any chance? So someone was saying that, hello, it is a recorded lecture. This is not a recorded lecture. I am live right over here and I'm so sorry. I was actually in an official meeting, which is why I could not come over here. But my children, I am back here for just you guys. And I hope I am audible. Ganesh, dear, I am so sorry. And also Zohin Talikoti, hello dear. So everyone, I am audible, I am visible is what I am assuming. Good evening, dear. Sri Raksha is what I am able to pronounce. Yes. Hi, Sri Raksha. Hello, children. Really, I'm sorry. I hope you guys forgive me. And I hope you guys are super ready for today's session, where today we are going to deal with two very important chapters. One is Principles of Inheritance and Variation, and the other one is Molecular Basis of Inheritance, both of which I had given you the masterstroke conceptual video. I hope you guys watched the one shot and you are here for solving the questions with me. Good evening, Sanjeevni. Good evening, Saima. Lots of love for all my little champions out there. So let's begin. I do not want to waste your time anymore now. Yes, you're practically visible and clearly audibly. Thank you. Okay, that's a very nice, what do you call, way of putting that. Okay, so here we have the first question and you have to give me the answer like this. Question number 1, A, B, C, D. Okay. So which of the following statements are correct with reference to prokaryotic genome? A. Monocystronic structural genes. B. Introns absent in structural genes. Transcription, translation are coupled processes. Primary transcript undergoes splicing. Only one RNA polymerase is present. So you have to tell me out of this, which ones are actually correct, and your options are from here. So you have to choose from here. Remember, you are being asked about prokaryotic genome. So just talk about prokaryotic genome, okay? Manoj, good evening. Voice of Zukrullah, good evening. That's a pretty name out there. Yes. So I see a lot of Josh in here today. Is it because the two chapters are really important? Okay. So all my dear children, please do not be shy. Give me answers, even if it is wrong. Listen, I am not the Dracula teacher out there who will drink your blood if at all you do not give me correct answers. So just give me just give me answers, and if it's not correct, I'll make sure I understand and make you understand that. Yeah. I'm waiting for your answers, everyone. Come on. Sri Raksha and Sanjeevni and Saima and Voice of Zukrullah. I hope you have another name as well. Okay, Sanjeevni says the answer for this question is B. B is what? Only BCE is correct. Only BCE is correct. Okay, I have got one vote for this. What about others? So only Sanjeevni has studied molecular basis of inheritance and principles of inheritance and variation. Hi Kaveri, is it like that? Children, if at all you do not talk to me, it's going to be really boring. Okay, so you have to talk to me, then only this video can be interesting and also you will learn with me. So I'll go with Sanjeevni's words since she is saying 1B is the answer and everyone else is just Chumma. Yeah, Sri Raksha is also saying B. Okay, that's the spirit. Okay, so B means B, C, E are correct. What does B, C, E? First of all, we're talking about prokaryotic genome. Prokaryotes means what? The ones which are very primitive in nature. Do they have nucleus? No, Baba. They do not have nucleus. So if at all this is a prokaryote, their genetic material is just lying around in one particular area which you call as nucleoid. Baba, do they have anything, any other membrane bound organelles out there? No. So they are very primitive. Okay. So first of all, it is written that they have monocystronic structural genes. Is that correct? Monocystronic means basically if I talk about the DNA and when I say the DNA transcribes into RNA, specifically mRNA, I mean that this particular segment is going to code for only one protein, which is when I say it that this particular thing is what? One gene is basically coding for one particular protein only. This is known as monocystronic condition. And in the case of Prokaryotes, they are not like that. Eukaryotes are like this. Prokaryotes are polycistronic. That means if this is a DNA, this DNA will be having multiple cistrons, and these cistrons will be coding for multiple proteins. So one protein, second protein, third protein. So P for prokaryotes and P for polycistronic. So this particular thing is wrong because here monocistronic has been mentioned. Now introns absent in structural genes. So what are introns? In your DNA, there are regions that code for proteins, which you call as exons, and there are regions which are just in between. They do not code for anything. They are known as introns. So this is a general case in the case of eukaryotes. But in the case of prokaryotes, do you think there are introns? No, Baba. There are no introns. So introns absent in structural genes. This is absolutely correct. So B statement is correct. What about C statement? Transcription and translation are coupled processes. So usually what happens is that in a normal cell, DNA to RNA as in transcription happens inside the nucleus. You're talking about eukaryotes. This is known as transcription. Then this RNA moves out to make proteins. That is known as translation. Both of them are two separate processes. It happens at different times. But in the case of prokaryotes, it does not have nucleus. So nothing is there to go in and go out. So basically both of them happen at the same position only. So you call this as coupled processes. They can happen simultaneously. So transcription and translation are coupled processes. Yes, that is a feature of what? Prokaryotes. Now primary transcript undergoes splicing. So listen to me. I told you from the DNA which has the introns, you get the RNA after transcription. So RNA will also have the introns. Now what do? You do not need the introns. They are not going to code for anything. So you have to remove this introns, and that process is known as splicing. But in the case of prokaryotes, Baba, they do not have introns. So what will you cut? There is nothing to cut out there. So primary transcript undergoes splicing. This would be wrong statement. Now only one RNA polymerase is there. To DNA to RNA formation, there is only one RNA in the case of prokaryotes, which is why we say this particular statement is also correct. So B, C, E is the only correct statement we have. That is why we say option B is correct. Hello dear. Kaveri ma'am, high weightage chapters for bio for KCET exam of 12th. I will definitely give a proper video for that, Kaveri. I hope that will help. Yeah. And Ankita gave me the correct answer, and Ananda is also here. Ananda also gave me the correct answer. Guys, you guys are awesome. The answer for this particular question is B. And I hope you have understood it. For everyone who had any sort of confusion, children, did you understand it? Or the confusion is still very strong? Ayyo, where did everyone go? So it's like one explanation and all of you are like Hello Ganesh. Ganesh says the answer for this question is B. You are right, Ganesh. Okay. So the most important chapter, Kaveri, generally is principles of inheritance and variation, molecular basis of inheritance and variation. Then you have human health and diseases, human reproduction. Generally, it follows the same trend as if you have for PU2. Just one thing is there, organisms and population also gives you enough number of questions. Okay? Understood. Thank you so much, guys. And also, can you do me a favor? Can you just give this particular video to all of your friends who are also studying biology? I really want to have every one of you watching this video. It is really nice when the numbers are really good. Okay? Yes, understood. Okay, let's go ahead with the next question. This question is easy. This is from principles. This is from principles of inheritance and variation. So I have taken in like alternate format. Okay. So identify which one of the given pair of option is correct with respect to Down syndrome and Turner syndrome. So here there are features of Down syndrome. Here there are features of Turner syndrome. You have to see both the features should be correct for each of the disease out there. Okay. Let's get it. So it's Jiri. Hi ma'am. Hello dear. Okay. So I think all of you have sent the video link to all of your friends, which is why people are coming in and joining. I'm really, really happy if at all that happens. Okay. Come on. Give me the answer for this. Your options are right over here. Read the options carefully and then only go ahead. So Down syndrome symptoms, short-statured individual, yes. In the case of Down syndrome, their height will be small. Gynecomastia in man. Turner syndrome basically means what? Turner syndrome means they do not have one X chromosome. There is only one X chromosome and they do not have the other one. This is XO condition. So my dear children, gynecomastia in man is seen in Klinefelter syndrome. Gynecomastia means formation of breast, right? And that is happening in man in the case of Klinefelter syndrome. So this option is wrong. So I've got one answer. Ankita says the answer for this question is C, and Srilaksha says the answer for this question is D. C and D is there. Multiple options are given to me. Okay, C and D. I'll lock this up for some time until and unless I get more votes. Sanjeevani also says it is D. So 2 votes for D. Okay, let me see. Okay. By the way, read the question very carefully. So let's see. Round head, partially open mouth. This is also a feature of Down syndrome. This is correct. Overall masculine development, and again seen in Klinefelter syndrome. They have overall masculine development, but gynecomastia, female breast, is also seen over there. So this is again wrong. Here you are asked about Turner syndrome. Now broad palm, physical and mental development retarded. This is a feature of Down syndrome. Sterile females with rudimentary ovaries. Sterile females, sterile means basically they are not fertile. They cannot produce babies. Why? Because, my dear children, they have rudimentary ovaries. Rudimentary ovaries means not well developed ovary, poorly developed ovary is there. If the ovary is not developed much, what is going to happen? Ova is not going to be formed properly. If ova is not there, do you think they can produce babies out there? No. That is why we say that they are sterile. Why is this happening? Because, Baba, only one X chromosome is there, other X chromosome is not there. So this particular feature is actually of Turner syndrome. So according to me, as of now, C should be the correct answer. But since a lot of you have said D, now Kavya Anand, they also said D. Let me check it out. Additional copy of X chromosome. In Down syndrome, can there be ever additional copy of X chromosome? Down syndrome means 21st chromosome additional copy is there, not X chromosome. It has nothing to do with this X chromosome. So this is wrong. Absence of X chromosome, yeah, this is correct. Absence of X chromosome, that is correct. So your answer, where both the answers are correct, is only C. So your answer for this question is C. Ankita is the only one who gave me the correct answer. Rest everyone did copy paste and somehow got the answer wrong. It is not D, it is C. Did you understand? We have to look for Down syndrome. We have to look for Turner syndrome. Why did you think D was an answer? If at all you saw this additional copy of X chromosome, do you think that can happen in Down syndrome, Baba? Ayyo. Ankita is happy. Only Ankita is happy. What about others? Others, it's okay, dear. This happens, okay? This happens. It's a part of life. We have to go through it. Wrong things happen. I hope you got it. Everyone give me a thumbs up if at all you got the understanding of why C is the answer. Everyone, quickly give me a thumbs up. Cute little thumbs up. And keep going, okay? Keep going. Let's see if at all it's possible for us to come back to form. I'm waiting for your thumbs up, then only I'll move forward. If you have any doubts, ask me now. And I have taken questions which from the concepts which have been repeated so many times so that you have an understanding. Okay, Sri Raksha gave a thumbs up. Thanks, dear. Okay, now I think I will go ahead. Next question is there. So only Sri Raksha understood. Ankita also gave me a thumbs up. Okay, Ananda said, okay, ma'am. Okay. So question number 3 is this. This is a very easy question. Which of the following enzyme increases the permeability of the bacterial cell to lactose? If at all, it's difficult for you. Sanjeevani also. Thank you, Ananda also. Fine. Listen, here the question is, this is a bacterial cell. Which one of these enzymes will make the cell membrane over here have increased permeability such that it can take lactose inside? So which is that enzyme that will be acting on the membrane's permeability? Everyone, Kavya says the answer for this question is B. Okay. Third, B. Dtech is their answer is say, he or she says the answer for this question is C. Trans-acetylase. Okay, Sanjeevani says B. What about others? So 2 votes for B. Kavya and Sanjeevani says B. What about others? Listen, the previous time we saw that everyone who said D got the wrong answer. So don't copy, okay? Try by yourself and then only tell. Okay, Ankita said A as an option, that is beta galactosidase. So you are giving me A, B, C, D option. So D only is remaining. If at all you give me D as the correct answer, that will also be done. So it's an MCQ question for me. Ayyo. Baba, it's very simple. Listen to me carefully. Permease is going to increase the permeability. The name itself is related to that only. So it is going to be permease. No. What does beta galactosidase do? Suppose this is an E. coli macha. Okay, this is an E. coli. this E. coli needs energy. How does it get energy? Basically, it is grown in a medium that is having lactose. So first of all, lactose has to get in. For that, you need the enzyme permease. Okay, now after it has got inside, this lactose has to be broken down to the energy molecule, that is glucose plus galactose. Who does that? It is basically a beta galactosidase who does that. Beta galactosidase. Now when it does that, obviously a lot of toxic substances are going to be formed, and there should be someone to deal with that. For that, you have the next enzyme, that is transacetylase. So basically 3 enzymes are there. Remember, Z, Y, A. Z codes for beta galactosidase, Y codes for permease, and A codes for transacetylase. This is Operon. Okay, so your answer for this question is going to be B, Baba. Increasing the permeability is B. With this, I understood one very important thing, that no one has watched the masterstroke conceptual video. Okay, so wait for a second and I'll be right back. Okay, just wait for a second. Guess. So honestly, you tell me how many of you scroll down like this and saw these 2 videos out there. Where is the video? Let me try to find it out first. That is molecular basis of inheritance. You see this? Molecular basis of inheritance. This is a one-shot video of the whole chapter. After watching this, there will not be any question that you won't be able to solve in both KCET as well as your PU2 examination. Second thing that is there. Second thing, principles of inheritance and variation, I did before that, and that is this. Principles of inheritance and variation. Please go watch this. You know what? Me, your ma'am, stood there just to make you understand for 2 hours. This is a 2-hour video that I shooted. Why? Is it because I have not taught you before? I had taught you. There is Sankalp videos that is there before in the month of what you call August only we did it. There were lots of things that were done. We redid it. Why? Thinking that it is something that is needed for you guys. If at all your concepts are not strong, you will not be able to do the questions. Understand that. Okay, here it's the ulta way around. Some people think if we solve the questions, we will get the same type of concepts out there. No, I'm telling you, it's going to put you in trap. Make sure your concepts are strong, then practice questions. Both of them are equally important. Got it? So those of you who have not watched it, promise me you're going to watch it, everyone, because I am pretty sure if at all you have watched it, there won't be this much confusion around it. Okay, Shabina, hello dear. Manjunath, hello. So I was not scolding you, okay? As a sister or as a friend, I'm just suggesting you one thing, that please make your concept strong. If not my video, then what's anyone's video out there? But please make it strong. Okay, done. So your answer for this question is permease. Fine, this is the answer. Now we go ahead with the next question out there and try to solve this. This is from Principles of Inheritance and Variation. A colorblind man marries a carrier woman. The percentage of their colorblind progeny in the next generation will be what? In the next generation will be what? A colorblind man marries a carrier woman. The percentage of their colorblind progeny in the next generation will be what? Guess. Ananda, yes ma'am. Okay. That itself is a good thing that, yeah, you are acknowledging to what I am telling you guys. But please take it seriously, okay? It's a very important thing. You have your examination. Okay. Malika, hello, hello, hello. Guys, can you try finding out the answer for this particular question? A colorblind, not from Google, okay? A colorblind man marries a carrier woman. The percentage of their colorblind progeny in the next generation will be what? Ananda says the answer for this question is B. I got a vote, that is for B. What about others? Brock Lensner. Okay, says the answer for this question is again B. Okay, that is 50%. Sanjini says D and Manjunath says B. I think maximum votes are for B. Listen to me carefully. Color blindness is what? It's an X-linked recessive disorder, right? Have that in your mind first of all. Now, you're talking about colorblind man. If a man has to be colorblind, his X chromosome has to be affected. Then only he will be colorblind. Now, he marries a carrier woman. Carrier woman means only her one chromosome is going to be affected. The other chromosome is still fine. It can overshadow the effect of that, which is why the female is not going to be colorblind. She is just going to be a carrier. She can give this particular chromosome. That is why we call it her as a carrier. Now let's try to fuse both of them. So over here, if this and this came, you got a female who is what do you call carrier? She is what? A carrier female. Okay. Now this and this, you got a female. You got a female who is what? Colorblind. So colorblind female. Mark this up. Okay. Colorblind female. Now this and this comes together, XY. He is a very normal male. He is a normal male. Now this and this comes up, you get X, C and Y. Over here, remember, if one chromosome FX is being affected, then they have to be affected. Why? Because there is no other X chromosome to overshadow the effect. So this guy is again going to be colorblind. So the colorblind man, or basically boy or male. So these two, out of 4 children, 2 of them are having color blindness. You are being asked about percentage of the colorblind progeny. You're not being asked about carriers, only color blindness. So 2 of them, so basically 2 out of 4 is 50%. So your answer for this question is going to be B. Yes. So those of you who said B, Ananda, I clearly see B, and Brox Lesnar also B, and Manjunath also B, and Kavya B, and yes, that's all. So I will come to this. Before that, let's move ahead with the next question, and that is question number 5. Question number 5 says, RNA polymerase 2 is responsible for transcription of. RNA polymerase 2 is responsible for transcription of. So I told you, in the case of prokaryotes, they just have one RNA polymerase. Remember, we just discussed that. But in the case of eukaryotes are like, oh my God, we need more and more. So they're not happy with single things, okay? So they have basically 3 types of RNA polymerase. RNA pol 1, RNA pol 2, RNA pol 3. So here you have been asked, RNA polymerase 2 is responsible for transcription of what thing? That is the question. Come on, come on. Try to give me the answer for this particular question. Hello, Flazing Annex out there. Hello, hello. Hello, Epic Gamers. So happy to see all of you guys out there. Okay, let's try to solve this question. I'm just waiting for you guys. Okay, Sri Raksha says the answer for this question is C. Is C. C is what? HNRNA. Okay, and then Flazing NX says C. Okay, to Manjunath says A. Ankita says C, and Ananda says C. Ganesh Patil, Sanjeevani, everyone says C is the answer. Yes, Baba, it is HNRNA. So basically, there are 3 RNA polymerase. Ayyo, where is it? 3 RNA polymerase. RNA pol 1, RNA pol 2, RNA pol 3. I'll start with this. 3 sounds like T. 3 and T is same. So basically RNA polymerase 3 is responsible for formation of tRNA, transcription of tRNA. Okay, now the primary function of RNA polymerase is to form rRNA, something similar to it. RNA polymerase 1, RR, everything is same. So rRNA, that is ribosomal RNA, is something that is being formed with the help of that. Now the second priority is given to form hnRNA. Heterogeneous nuclear RNA. You know, DNA, which undergoes transcription, it forms hnRNA first. This hnRNA undergoes some processing, and finally you get the final mRNA. So this is like a precursor for mRNA. So this is the flow that we have. But also remember, here, when I talk about rRNA, there are different types of rRNA. 28S, 18S, and 5.8S. These 3 types of rRNA are formed with the help of this. Okay, now over here, tRNA, I told you. Apart from that, here also one type of RNA is being formed, and that is known as 5S rRNA. 5S rRNA. This is something else. Apart from that, small nuclear RNA, that is also being formed with the help of what? With the help of RNA polymerase 3. So please remember, if at all you remember this, you might be able to solve any question out there. Got it? Okay. So I think almost every one of you gave me the answer as C, and those who didn't retracted their answer also. Okay, Fine. Now question number 6 is, when a single gene exhibits multiple phenotypic expression, the phenomenon is called. This is principles of inheritance and variation. So I'll read out the names of the children who gave me the correct answer. By then you gave me the answer for this. Sri Raksha, Flazing, Anix, and Ankita, Ananda, Ganesh Patil, Sanjeevani, Malika, and yes. Ma'am, will I'll see Master Stroke to get good marks in PU2 boards? Definitely. Definitely you will. Ma'am, could you please explain again? Definitely, I can explain again. So children, just a second, I'll go explain that and I come back again because I think it's important for everyone, right? And you saw the answer, right? Tell me you didn't see the answer. Listen, you have 3 RNA polymerase, RNA pol 1, 2 and 3. This is only found in the case of eukaryotes. RNA polymerase 1 is responsible for transcription of rRNA, okay? That is ribosomal RNA. That is something that is going to build the ribosome out there. So rRNA again are 3 things: 28S subunit, 18S and also 5.8S. All of them ends with 888. 28S, 18S and 5.8S. Done. Now over here, RNA polymerase 2 is for transcription of hnRNA. hnRNA is a basic thing that we need to make proteins. So DNA transcribes into hnRNA, that is heterogeneous nuclear RNA, and this RNA undergoes some processing and finally we get our mRNA. So this is the second priority. Now third priority, third is there, third, right? Third priority is given to form the tRNA. Okay, and along with that, some type of rRNA is also being formed, and that is known as 5S rRNA. 5S rRNA. Now, apart from that, small nuclear RNA is also something that comes up in this particular group. I hope this is clear now. Okay, so which is why your answer was C. Got it? Everyone, give me a thumbs up if at all you got it. This is important. You can get questions related to it. Ananda, give me a thumbs up. Thank you. Answer was C. Ganesh, forget the answer. I'll show once again and I'll ask you the question, okay? Okay. Epic Gamer as well as Flazing Anix, is it clear to you, bacha log? Is it clear? Shall I move ahead? Thank you, ma'am. Understood. Okay. Thanks to you too. Okay. Now, I know you know the answer, but guys, you have to still try. So when a single gene exhibits multiple phenotypic expression, this phenomenon is known as polygenic inheritance, incomplete dominance, pleiotropy, codominance. And since I showed you the answer by mistake, I'll ask you another question with the same help of this. Hi, Numan. Okay. Guys, what do you think is the answer? And thank you everyone for giving me the thumbs up. And I see 17 children watching this video. Proud of you guys. Okay. So A, Numan says A, Manjunath says C, Sri Raksha also says C, Malika says C. Okay, this is actually known as pleiotropy. What happens basically? Mendel Baba said that one gene codes for one character. No, nothing apart from that. But it was later found that one gene can actually code for multiple characters, can affect multiple characters. And this is known as pleiotropy. You saw in phenylketonuria. I was about to ask a question related to that and I gave you the answer. Okay, so tell me, in phenylketonuria, what are the 2 things that are being affected because one gene is being mutated? What are the 2 things? For example, phenylketonuria is caused because phenylalanine hydroxylase is not being formed. Phenylalanine hydroxylase is not being formed. But if this gene is not being formed, what are the 2 effects that are being seen? Any guesses? You have to tell me the effects, both of the effects. First effect, second effect. So start with the first one. What is the first effect that is being seen? Come on. So you saw the answer, which is why you are being asked one more extra question. I'm the toxic teacher that you have. I'm just kidding, okay? I'm not toxic. Children, I am waiting. So when I said there are 17 children watching this video, all of them ran away, I think. And now, come on guys, show some spirit. The first thing that is getting affected when you do not have phenylalanine hydroxylase is what? Is this going to be? Is your slope so? Loss of skin pigmentation. Ishant is there. Hello, dear. That's so sweet of you. Yes. So you see something that is known as hypopigmentation. Okay. You see something that is known as hypopigmentation. That means the color of our hair as well as our skin, it is going to go down. It is going to go down. That is known as hypopigmentation. In your NCERT, it is written as reduced hair and skin pigmentation. It's not like your hair is getting reduced, only the color of it is going to get reduced. That is what you mean by reduced hair and skin pigmentation. Apart from that, when phenylalanine hydroxylase is not being produced, the major function of this guy is to convert phenylalanine to tyrosine. So basically this conversion is not going to happen. If this conversion is not happening, this thing, phenylalanine, is going to accumulate in our body in the form of phenyl pyruvic acid. And when it accumulates in our body in the form of phenyl pyruvic acid, what happens, my dear children? There are chances that it can lead to mental retardation. So basically, over here, this is one feature that is affected. As I told you, the second feature that is there, it is hypopigmentation. Why does it happen? Because tyrosine is not being formed, and tyrosine is super important for the formation of some other pigment that is responsible for our skin and hair pigmentation. If this pigment is not there, basically leads to hypopigmentation. That is why we say that phenylketonuria is an example of pleiotropy. Got it? Everyone who heard me. Point mutation or frame shift mutation. Numan, it's not given in your NCERT. Even I have to check that. Okay. So in your NCERT for point mutation, only sickle cell anemia is given. I have to check what type of mutation causes this. But anyway, yes. Got it? The concept. If you got it, everyone, let's move on to the next thing. Affects the individual lack an enzyme that converts amino acid phenylalanine into tyrosine. Exactly, Ananda. Okay, so Manjunath also gave me thumbs up, and I think I am good to go with the next question out there. Yes? Okay. Mallikarjun also gave me thumbs up. Fine. Question number 7. The mRNA has untranslated regions. Sorry, Mallikarjun, I called you. I'm so sorry. It's Mallika. Okay, so that was an imaginary character. Yes, let's try this. The mRNA has untranslated regions at 3' and beyond the terminator codon, at 5' and before AUG, at both 3' and 5' and beyond terminator and before AUG respectively, and AUG and terminal codon flanks the UTR. What do you think is the answer for this particular question? A, I guess. Okay. A is something that you're guessing. What about others, children? You have been asked about the untranslated regions of mRNA. So you have to be a bit careful around here. A. Anand is also there. Hello, Anand. Okay. Keep on guessing. But also think, listen, this is the mRNA. You know the direction of mRNA? mRNA is from 5' to 3' direction. At the 5' position, it has the initiator codon, which is known as AUG. At the 3' end, it has the stop codon. It has what? The stop codon, or you also call this as the terminator codon, because here it is going to terminate the process of translation. Now Ankita says the answer for this question is C. Manjunath also, Manjunath A, Ananda C, Ankita C, and Ananda C again. Kachappa is there. Hello dear, says the answer for this question is A. Okay, so basically I'm going to decode the answer. Listen to me carefully. This is the mRNA where you have the initiator as well as the terminator codon. The UTR, untranslated regions. So this region is going to undergo translation for sure. You know that. Before that, you have UTR. After that, you have UTR. UTR stands for untranslated region. They are not going to undergo translation, right? So can I say it is before the 5' end? So it is basically before AUG respectively. Before AUG means before the 5' end. And also beyond the terminator codon. Is it beyond the terminator codon? Yes. So basically your answer for this question is going to be C, right? Isn't it going to be C? So at both 3' end, that is that, and 5' end, beyond the terminator codon out there and also before the AUG respectively? Yes, yes. So which is why your answer for this question is going to be C. At 3' end beyond the terminator codon, at 3' end beyond the terminator codon, that is correct. This is correct. At 5' end before AUG, this is also correct. But this option has both of them together, which is why this will be definitely correct. AUG and terminator codon flanks the UTR, that is not going to be correct. What is the use of UTR? UTR, untranslated regions, provide stability to the mRNA. It helps in initiation of the process of translation. Very, very important. This is a frequent PU2 question as well. What is the function of UTR? What does it do? It provides stability, and also, my dear children, it helps in initiation translation. Okay? Kaizuko, hello, nice name. Ma'am, we get HOTS question from transcription and in practice. Could you please summarize it as it is related to the question itself? So, transcription here. This is translation, by the way. Okay, so do you want it right now? I can basically. But I do not exactly have the question. So, I can just say, always remember you have the 3' and the 5' here, and the 5' and the 3' here. Okay, now I'm going to make the transcription in it. Here you will be having the promoter sequence, here you will be having the terminator sequence out there. This strand which is there, it is the template strand. And the opposite strand is going to be what? The coding strand. Template strand means the one that is undergoing transcription. So you might be given this strand and you might be told, make a transcription in it. So how do you do that? Suppose over here they have mentioned it as A, T, G, C and things like that. Make a complementary strand over here with the opposite thing. So A goes with T, T goes with A, G with C and C with G. And you have to make the whole thing. This is how you make the transcription in it, and make sure you're writing the promoter here and you're writing the terminator there, and then they might ask you show transcription. How do you show transcription? So basically this particular strand which is there, this strand which is there, it is going to undergo transcription, right? Template strand undergoes transcription. So basically here you make the complementary mRNA strand for it, or basically just RNA strand for it. How will it look? It will look exactly like this. That is the what do you call complementary strand that you made over here. Just that you have to replace the T with U, because in the RNA you do not have T. So do not try to make the whole thing one by one again. Instead, whatever coding strand you have made over here, just replace the T with U and you have got the new what do you call transcription, what do you call mRNA strand out there. That's all. Okay? Numan, so dear, I was thinking I'll do a HOTS video separately because I do not know exactly what question is there that you are asking me as of now. But there will be a separate video where I'll be discussing more about it. HOTS only. Okay? High order thinking question. And Numan, I hope you understood at least this much. This was like a least of thing that I could do for you as of now. Yeah? Yes. Thank you, Numan. Okay. Next question, guys. Question number 8. In a dihybrid cross between a true breeding round yellow seeded and true breeding wrinkled green seeded pea plant, the ratio of segregation of round and wrinkled seeded traits in F2 generation is what? Guess. In a dihybrid cross, you're talking about dihybrid cross. Why dihybrid? Because you're considering two characters at once over here. What are the two characters? Round and yellow. Round is for shape and yellow is for color of the seed. So these are two characters that you're considering together. So round and yellow you're considering, and also you're considering wrinkled and green plant. You are crossing this with this. Here also this is the shape and this is the color. Now you have been asked this question that tell me the ratio segregation of round and wrinkled. You're not asked about the color, you're just asked about the shape. So what will be your answer for this question? Ananda says it is C, Manjunath says it is B, and Ananda says it is C. Manjunath says it is B. Manjunath says it is B, and the answer for this question is going to be B. Why B? Because, Baba, listen to me. We know the ratio, that is 9:3:3:1. Do you guys know how to remember the ratio? It's very simple. 9 of them are going to show the dominant character. Round and yellow are the dominant character when you compare it with wrinkled and green. Okay, so this is going to be round and yellow. Cool. Now, one which is there, it is going to be the recessive character that we have studied, and that is wrinkled and green. Wrinkled and green. Now here three are there. What is this? This is the non-parental character. That's the new characters that are there. For example, here round was with yellow. This time we will make round with green. Okay. Here wrinkled was with green. This time we will make wrinkled with yellow. Now you check how many of them are round here and how many of them are wrinkled here. So I see 9 of them are going to be round, and 3 of them are also going to be round. 9 plus 3 is equals to what? 9 plus 3 is equals to 12. Wrinkled, if I see, my dear children, 3 plus 1. That is 4 of them are going to be wrinkled. 4 of them are going to be wrinkled. So 12 ratio 4 is equals to what? Can I say it is 3:1? Can I say it is 3:1? Yes. So which is why your answer is this. But did you know you need to do all this drama out there? No. If at all you just consider monohybrid cross, capital T and small t. We have studied that, right? Which one is the dominant one? That will be having, that will be showing up, what do you call, thrice in F2 generation. 3 of them are going to be tall and 1 of them are going to be dwarf. Similarly, you just consider round and green over here. Round is the dominant thing, so round will appear 3 times and wrinkle will appear only once. So 3:1. Okay, did you get it? So this is a dihybrid based question, but at the end you are being asked about the monohybrid cross ratio, because you are only considering one thing, and that is shape. Only one character is being considered. Now if that is not easy for you, then you can do this way also. Okay, did you understand? Everyone who is watching this video and who all have retracted your answers. Oh my God. So Manjunath gave me the correct answer. It was 8th question answer was B. And I hope everyone understood. Also, one more thing, you need not retract your answer. Let me tell you, do you think any other person over here knows you? Are we all world famous celebrities? Is it okay if at all we do mistakes? Perfectly okay. Is it the major examination? No. Are you learning? The answer is yes. So definitely you have to keep going. It's not a problem even if you give wrong answer. Okay, do not take it to your head. Fine. Ananda also gave me thumbs up. So now we'll go on to the next question, that is question number 9. This is related to molecular basis of inheritance, and a very nice experiment. If at all you remember my Puchu story, you will be able to crack it up. Following representation P, Q and R denote few steps of Griffith experiment. Identify the correct step once. Okay, so these are the few steps that are given. You have to identify which one of them are the correct step which were found in Griffith's experiment. R strain, when it was injected into the mice died. Is it? S strain, heat killed one. When injected into the mice died. R strain, when injected into the mice lived. Okay. What do you think is the answer for this question? You have options starting from here to there. Read the answer question carefully and then only answer. This is 2 recent questions. This is 2024 question out there. Okay, Ananda says the answer for this question is B, that is R only. That is only this. Okay. What about others? Shivaraksha says B, Ananda says 9th B, Manjunath says B. Okay, B. Kachappa also says B. Okay. Kachappa, that's a very cute name that you have, dear. B. So everyone says B, which is why I'll log B. Yes, the answer for this question is B. Why? Because Streptococcus pneumoniae has 2 strains. One is R strain, one is S strain. R strain is a safe strain. It does not cause any death. S strain, which is there, it has a smooth covering around it, mucus polysaccharide covering, which makes it virulent. If at all any organism is infected with this, organism will anyway die. Okay, so that is a problem. So R strain, if at all you inject in the mice will live. Puchu is not going to die. The name Puchu is basically my mouse. Okay. Now S strain, heat killed. That means I am treating the S strain with heat. That means anything that was causing trouble is not there now. So it is safe now. So S strain, which is heat killed, if I inject into the mice will live. If at all I am just directly putting normal S strain, the mice will die. But here I am treating with heat, so obviously mice is not going to die. But here it is written, it dies. R strain injected into the mice live. So which is why your only answer for this question is question number B. Kachappa, my name is Suma. Oh my God, that was actually a cuter name, but still yes. Okay, I will try to call you Suma from now. Okay, question number 10. Question number 10, everyone. And all of you, Anand also gave me the correct answer. Good. All of you good. So find the incorrect statement among the following. Try to find the incorrect statement among the following. In sex-linked recessive traits, the gene is transmitted from unaffected carrier female to some of the male progeny. Sex-linked recessive traits. Just imagine any sex-linked recessive trait. For example, color blindness. So here it is said that the gene is transmitted from unaffected carrier female to some of the male progeny. What do you think? You have to find the incorrect statement. So yes, if she is a carrier, and males definitely are formed of two chromosomes X and Y. Y chromosome they get from the father, and the X chromosome they get from the mother. So yes, it can be transmitted to some of the male progenies. The carrier can transmit. So this is the correct answer. I mean a correct option. You have to find what is wrong, right? Manjunath says the answer for this question is C. Let me see. So B says accumulation of phenyl pyruvic acid in the brain results in mental retardation. Remember we studied about what do you call phenylketonuria, and I told you in phenylketonuria conversion does not happen of phenylalanine to tyrosine. If phenylalanine is not converted, it is going to get accumulated in the form of what? Phenyl pyruvic acid, and it actually causes mental retardation. So this is also correct one. Still now we have not spotted the wrong one. Sri Raksha says the answer for this question is C. Ananda also says it's C. Okay, since there are so many people who are now saying it is C, let me check it out. Individuals affected by Down syndrome will have congenital heart disease, as in genetic heart disease, and are more intelligent. Now we all know that people who have Down syndrome will be showing symptoms of mental retardation. So over here intelligence is being mentioned, which is why this particular statement would be wrong, and you have to be really careful, because until here it is all correct, here it is going to get wrong. Now Turner syndrome is caused due to absence of one X chromosome. Yes, and this condition is XO condition, right? One X chromosome is not there. So this is also correct. So all of you who gave me the answer as C, awesome. Okay, Ankita also gave me the answer as C. Awesome, awesome, awesome. So yes, now my dear children are getting back into track. Now this is a question that we discussed before in a different format, which is why I have a little expectation from you guys that you give me the correct answer. Think for this question carefully and then only go ahead with giving me the answer. Okay, so the question goes like this. Eukaryotic genes are monocystronic, but they are split genes because eukaryotic genes are monocystronic, as in basically if I talk about the DNA, if a gene is there, a gene will be having only one thing, a cistron only, that will code for only one protein. So, but still they are known as split genes. What is the answer for this question? You can give me the answer in this format, 11ABCD. 11ABCD. Because I will not know if at all you are repeating the previous question or not. Sri Raksha says the answer for this question is B. Okay. What about others? What about others? Eukaryotic genes are monocystronic, but they are split genes because this is 2023 question. Still thinking. Probably because I have put expectations on you guys. Is it because of that? Okay. Ankita says the answer for this question is D. Okay. Listen. Why do you call them split genes? I told you, eukaryotes, my dear children, they have exons, they have introns. Again, they have exons, they have introns. This whole thing is nothing but a gene. And this gene is going to code for only one protein. But only these sequences are going to be of any use. Introns are there and they are of no use out there. So can I say they contain introns only? That will be wrong. They contain exons also. Exons are interrupted by introns. Yes, exons are actually interrupted by introns out there. That is the reason why you call them as split genes. Introns are interrupted with mutons. That is not given any anxiety. Why should you be bothered about that? They contain exons only. That also cannot be correct. So your only answer for this question is B. And the first person who gave me this answer was Sri Raksha. Okay. And after that, Ankita and Ananda also have now given me the answer as, hey, wait, Suma also gave me the correct answer. Yes. Okay. Manjunath, just check once, okay, Macha? Everything is sorted. So this particular question is clear, everyone? Just give me a quick thumbs up if at all it is clear. Understood? If understood, if clear, just give me a quick thumbs up. Okay, thank you, thank you, thank you, thank you, thank you guys. Okay, now test cross in pea plant is what? Test cross in pea plant is a cross between F2 tall plant and a recessive parent, a cross between F2 dwarf plant and a recessive parent, a cross between F2 tall plant with a dominant parent, a cross between two F1 plants. What do you think is the answer for this question? And thank you for giving me thumbs up. Sri Raksha, Ananda, Ankita, Manjunath and also Suma. Okay, guys. Hello, 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 hello. Yes. Hello, children. Am I audible? Hello, hello, hello. Am I audible now? You know what happened? The battery got ended. That much talking I do literally every single day. The battery got ended. You see? Am I audible? Clear? Give me a quick thumbs up. Yes, okay. Finally, yes ma'am, audible. So I just went to change the battery up there. Now I think it's fine. Okay, so where were we? Okay, we were here. So I think, hello. Okay, so I think we were here. So plant was seeming tall for me. Now I do not know whether it's tall like this or whether it's tall like this. That is homozygous tall or heterozygous tall. So what do we have to do? We have to cross it and see. By which plant we are going to cross it? We are going to cross it with a recessive parent plant. That means something which is like this. Both of them are going to be crossed. Why? Because we need to find it out properly. Because we want pure tall like this. Then only the progenies will also be tall. So what are you going to do basically, my dear children? Let's do the test cross. So the test cross if I cross this with this, I find that all of them, whatever you do, are going to be tall only. Why? Because at least one capital T is going to be there. So my result is all of them are tall. Over here, if I do the same thing, my dear children, what is the thing that I'm going to find out? It's small t here, and it is capital T small t here. This and this, I get capital T small t, this and this capital T small t, this and this small t, and this and this small t small t. Two of them are definitely tall, but two of them are going to be dwarf. So with this, I get an understanding that, okay, my plant was actually, if at all my result is like this, then the plant is like this. If my results are like that, then the plant is going to be like this. So this is known as test cross. B is not the correct answer, children. A is the answer. A cross between F2 tall plant, this is for second generation tall plants, and the recessive parent, then only you will get an understanding what is this particular plant tall like. Is it like this or is it like this? Did you get an understanding of it, or are you still stuck with B option? Come on. 12A is the answer, okay? Everyone who is watching this video and who has even a little bit of confusion, please ask me before going ahead. If you have not understood this, super important video. Super important question and also super important video. Okay? If you got it, can you just give me a quick thumbs up? Children? If at all you cross with capital T, what is going to happen? Capital T will is a dominant thing. It will show up. There won't be any chance that you can get dwarf plants then. That is why you specifically cross it with a recessive parent plant. Okay, there is a reason you cross it with a recessive parent plant. Okay, I think the video buffering is going a little slow. Okay. Okay, Sri Raksha, thank you Ananda and Malika and also Ankita. Okay, cool. Let's go ahead then. Let's see the next question. This question is really simple. This is from Molecular Basis of Inheritance. Came in the year 2023. Let's ace it. Histone proteins are positively charged because they are rich in basic amino acid residues such as what? Arginine, lysine, arginine, phenylalanine, arginine, proline, and arginine, alanine. What do you think is the answer for this particular question? Histone proteins is rich in what amino acid? That is the question. Okay, Preetham says the answer for this question is A, Sri Raksha says A, Suma says A, Anand also says A, Ananda also says A. Since you guys are saying A, I will lock A. The answer actually is arginine and alanine. So you can remember like A and L, or you can remember like this, Los Angeles. Okay, Los Angeles. So both of these two very important amino acids are there inside what? The histone proteins. And your DNA is negatively charged. So negative positive comes, which is why your DNA is able to wrap around the histone proteins, which are rich in these positive things out there. Okay, got it? Fine. Now going ahead with the next question out there, and that is question number 14, which says the gene for hemophilia is located on X chromosome, hence it is normally impossible for a dash. For a dash. Guys, the answer for this, the gene for hemophilia is located on X chromosome. Hence, it is normally impossible for what? Indra Power is there. Hello, dear. Indra Power says the answer for this question is A. Is it this question that you're talking about? Or is it some other question? A is what? Hemophilic father to pass the gene to his daughter. Preetham says the answer for this question is D. Okay. What about others? Sri Raksha also says D. Anand also says D. Okay. Let's try to decode then. Hemophilia is located on X chromosome. We know that. Hence, it is normally impossible for a hemophilic father to pass the gene to his daughter. Listen. If a father has hemophilia, the condition is going to be like this. A daughter is made up of 2 X chromosome. So yes, he can pass the X chromosome out there. So yes, he can pass the X chromosome to the daughter. So this is wrong anyways. You are, this is actually possible, right? You are being asked about what is impossible. So A is definitely possible. Carrier mother to pass the gene to her daughter. Carrier mother, my dear children, again, carrier mother means one of the chromosome is affected. Can she pass it to the doctor? Yes, she can pass it to the daughter out there. So this is also possible. Carrier mother to pass the gene to her son. Son also has XY chromosome, and the X chromosome comes from the mother itself. So yes, it is possible for that also. Hemophilic father to pass the gene to his son. Listen, hemophilic father means it is a condition like this. Can he pass the gene to his son? Son is going to be formed of X and Y chromosome. X chromosome they get from the mother, Y chromosome they get from the father. So father cannot pass this chromosome to the child, that is the son, my dear children. Only a mother can pass X chromosome. So it is normally very much impossible for a hemophilic father to pass the gene to his son. Always understand that. So you cannot see men-to-men transmission in the case of X-linked recessive disorder until and unless female is not affected. Okay, so your answer for this question is Baba D. So let me see who all gave me the correct answer. Okay, Preetam gave me the correct answer, Sri Raksha gave me the correct answer, Anand and Suma gave me the correct answer, and Ganesh Patil also gave me the correct answer. For everyone who had given me some other answer, do not be worried, things happen. Genetics is a bit complicated, but you will be able to crack it up. Okay, done? Everyone, can you give me a thumbs up if everything is all cool, everything is working well? Shall we go ahead with the next question then? We are on the 14th question. Okay, thank you. So I think it's getting loaded a bit late over here. Now next question. During transcription, the DNA strand with 3' to 5' polarity of the structural gene always acts as a template because what? So during transcription, the DNA strand with this polarity, that is 3' to 5', it acts like a template strand, as in the one that is undergoing transcription. Why do you think that is happening? Why do you think that happens? Sunita, hello dear. Hi, hi, hi. Okay, so first option is enzyme DNA dependent RNA polymerase always catalyze the polymerization in 3 to 5 direction. Is it? Is RNA formed in 3 to 5 direction? I thought it was 5 to 3 direction. But anyway, let's see. Enzyme DNA dependent RNA polymerase always catalyze polymerization in both direction. That is a definitely very big news. Okay, Sunita says the answer for this question is D. Okay, came up with a banger out there. Okay, nucleotides of DNA strand with 5' to 3' are transferred to mRNA. Okay, is it? Enzyme DNA dependent RNA polymerase always catalyze polymerization in 5' to 3 direction. Baba, this is your answer. D1 is the answer. If this is a template strand, a new strand is going to be formed. In what direction? The opposite direction. So this will be 5' to 3'. Your DNA polymerase, your RNA polymerase, even the reading of the mRNA in translation, everything happens in 5' to 3' direction only. The strands which are going to be transcribed or replicated will be in 3' to 5'. The new strands that are going to be formed always will be in 5' to 3'. Always remember that, and that is exactly given over here, which is why your answer for this question is going to be D, Macha. Okay. Yes. So Ganesh also correct, Ananda also correct, Anand also correct. What about other children? My dear Suma as well as Sri Raksha and Preetam and Indra Power. Great going, great going. Okay. I hope you guys understood. Now we have next question, that is question number 16. From the following pedigree chart of a family, one, you have to choose one of these things. What exactly is this pedigree about? It's an autosomal dominant trait, it is an autosomal recessive trait, it's an allosomal dominant trait. Allosomal means sex linked, okay? And it's an allosomal recessive trait. What do you think is the answer for this question? I hope you can see the pedigree clearly. Yeah? What do you think is the answer? Sunita says the answer for this question is A. 16th answer is A. What about others? Do you have any different answers? Dhanush is back and Dhanush says the answer for this question is B. Okay. And still waiting for others. Ganesh Patil also says B and. Sri Raksha also says B and Suma also says B. Yeah, now my older children also coming. B is what basically autosomal recessive trait. Listen to me carefully. So if at all you see over here in the pedigree chart, this and this basically means that the person is affected, right? Do you see that in every single generation a person is affected? No. Over here you see none of the parents are affected, but the children are. So you see skipping of generation. Whenever you see skipping of generation, just understand it cannot be a dominant disorder. Whenever you see skipping of generation, you cannot say it's a dominant disorder. Rather, you will say it's a recessive disorder. So now we have to eliminate two options which says it is dominant. This and this is definitely gone. Now we have to find whether it is autosomal or sex-linked. So autosomal one, my dear children, will be equally affecting the male and the females. In sex-linked, it will be mostly affecting what? The females out there. Just a second. So you got the answer. Just a second. Now, come on. So today nothing is working well, and I think it's because, okay, just give me a quick second. Good things take time, yes, and it took time. So over here, males and females are equally being affected, male and female in one generation. Here also, okay, one female is affected. But in the case of allosomal, you will see more males being affected, which is why your only answer for this question is going to be B. Okay, your only answer for this question is going to be B. And I see a lot of you gave me the correct answer. It's okay, Sunita. No worries, dear. No worries. It happens. I always tell you. By the way, everyone who is watching this video, are you liking this lecture? If at all, yes, what do you have to do? Click on the like button and share it with your friends as well. Okay, so DNA replicates semi-conservatively was first shown in what? DNA replicates semi-conservatively was first shown in what? So someone saw the question even before. Okay, Preetham says B, Sri Raksha says B, Dhanush also says B. The challenge is going on strong. Semi-conservatively means what? Suppose this is a parental DNA, okay? Now, if you want to make the new DNA, what do? Do you make the new thing altogether? New only? No. Basically, the strands are going to separate. Now, we got one strand over here, you got one strand over here. Then you get the new strand over here, new strand over here. So, basically, if at all you see, one strand is from the parental strand only. So, this is semi-conservation. Entirely, you're not getting something very new. Half of it is from the parents itself. That is known as semi-conservative mode of replication. And I see Sunita, Ganesh and Suma also gave me the correct answer. The answer for this question is what? B. Escherichia coli. You also call this as E. coli. Awesome, children. Awesome, awesome, awesome. DNA replication was first shown to be semi-conservative in E. coli by muscles and installer. What? Ganesh? I think there is some very big trouble with, what do you call that as? I forgot. Autocorrect. Sunita, yes, I remember. Yes, I definitely knew that you had a different name, but I forgot the other name, dear. I'm so sorry. Yes, Jessica. Okay, so cool, dear. Let's go ahead with question number 18. Match the column 1 with column 2. Here are your options and here are the other options given. Carefully go ahead with each of them and do not think that it is not there in the NCERT. Allosome is not there in the NCERT. Allosome means sex chromosome linked things. Okay? Allosomes means sex chromosome. Your X and Y chromosomes are allosomes. So accordingly, you have to give me the answer for this particular question. You have options from here to here. Go ahead, read the options carefully. Whenever you are dealing with a match the following question, first of all, read one column fully, then go ahead, read fully the second column. Ganesh, typing mistake, it's okay, dear. Okay. Preetham says the answer for this question is D. Okay, what about others? Okay, sorry. Preetham says D, Dhanush says D, and Jessica says D, and Sri Raksha says D, and yes, everyone says D. D is what? This. Let me see if at all it is D. Autosomal trisomy. Trisomy means one extra chromosome is coming. and that is seen in the case of Down syndrome. Is that so? Yes. So how many autosomes do we have, dear children? We have 22 pairs of autosome. The 23rd pair is basically the sex chromosome out there. So 21st chromosome, if at all we get an extra copy of it, for example, you just need to have 2, but you got 1 more. This is known as autosomal trisomy. That happens in Down syndrome. Now B is what? B is allosomal trisomy. Sex chromosome, 1 more extra chromosome is joining up. Allosomal trisomy, it is seen in Klinefelter syndrome. You needed to have XY chromosome, but now you have 1 more X chromosome. That is why it is known as allosomal trisomy. Okay, allosomal monosomy, sex chromosome, only 1 is there. Allosomal monosomy means loss of 1 sex chromosome. That is seen in Turner syndrome. There is only 1 X chromosome, other X chromosome is missing. So this goes with this. Now cystic fibrosis. Cystic fibrosis, my dear children, is given just in your NCERT. You have to read it carefully. Cystic fibrosis is basically a Mendelian disorder and actually an autosomal recessive Mendelian disorder. It's not dominant. Now, so your answer for this question is actually D. Now I want to ask everyone a single question, okay? And let me see who gives me the correct answer for this. Which one is the only autosomal dominant disorder which is given in your NCERT? Which one is the only autosomal dominant disorder which is given in your NCERT? That is the question. Now everyone try to give me the answer for that. If at all you know, you can try to give me the answer for that particular question. This is like, I'm just giving it to you guys because I really want you guys to know the basics of NCERT. It can pop up anytime. Which one is that autosomal dominant disorder, which is the only thing that is mentioned? Myotonic dystrophy. Yes, Preetham, Kit Kat for you whenever we meet, okay? Jessica, I think there is a typo out there. Okay, it's very funny how basically autocorrect, or basically when you type very fast, it comes out to be in different. So it is Moroni dystrophy. It's a new disease that Jessica has found. But yes, good efforts out there. I am pretty sure you meant myotonic dystrophy. Yes. So let's go ahead with the next thing that we have, and that is question number 19. Which aspect forms the basis of DNA fingerprinting? Which aspects forms the basis of DNA fingerprinting, my dear children? The satellite DNA showing high degree of repetition in DNA segments, the ratio of purines and pyrimidines present in DNA, the amount of DNA found in the samples of blood, saliva and skin, the sequence of DNA present at the ridges and the grooves of the fingerprints. What do you think is the answer for this question? Which aspect forms the basis of DNA fingerprinting? And you have to choose the most correct one. Okay, you might feel that one is correct, the other one is also correct. Most correct one has to be chosen, okay? Ganesh Patil, yes dear, it's myotonic dystrophy. So over here I see Jessica gave me the answer for this question, that is A. Dhanush also gave me the answer for this question, that is A. The satellite DNA showing high degree of repetition. Yes, so we are 99.9% same, but there is 1%, 0.1% difference, and that 0.1% difference is basically because of few very important things, that is the differences that is seen in our DNA, which are satellite DNA. So satellite DNA is basically repeated segments of DNA out there. You might have basically 10 of them, I might have 5 of them, and that basically makes us really different. And not just that, obviously, not just satellite DNA, there are other parts of the DNA also which codes for protein, which makes us different. But yes, a huge thing goes for the satellite DNA also. So which is why your answer for this question is going to be A. Amount of DNA found in the samples of blood, saliva and skin is not the answer. Your answer is A. Mostly we go ahead with the C option, but Baba, C is not the correct answer. Okay, it is A. It is definitely A. So those of you who have went with C, it's okay. It is generally a confusion that happens in children, but I hope it's clear now. Okay? The basis is this, not the amount of DNA that is found in here. Cool? Okay. Let's go ahead with question number 20. Now this is an important question, a very easy question, some concept that we have not dealt with in this lecture, and that is blood group. I have fun, really have fun while teaching this. So the genotype of a husband and a wife are IA, IB. the husband suppose is IAIB, and husband is IAIB and the wife is IAIO. Now you have been asked, among the blood types of the children, how many different genotypes and phenotypes are possible? How many genotypes and phenotypes are possible? Yes, Sri Raksha, the C option would be wrong, because over here the most relevant option has to be taken up. So it's not like the amount of DNA found in samples of blood and saliva, that is the basis. No, amount can be less, amount can be more. Even a little speck of DNA which is there, that would be more than enough. You can amplify it with PCR, right? So basically that is not the question. You are asked the basis of DNA fingerprinting, which is why A would be the answer. The basis is that how different are we in the repetitive sequences that we have. Okay? Okay, because that is something that we all share, and there are differences in that also. So yes, guys, give me the answer for this question. 20, your answer is B, is what Ganesh says. Suma says it is B, and Dhanush says it is B. Preetham also says it is B. Jessica also says it is B. Is it B? Let me check it out. Okay, so you have to find the number of genotypes as well as the phenotypes. So this and this goes, I get IA. In this case, the blood group is going to be A blood group. This and this goes, I get IAIO. Listen, when A and O comes, A is going to dominate over the O, which is why over here again it will be A blood group. This and this, IAIB. Over here, A and B, both of them are there. It shows co-dominance, which is why you get AB blood group. Finally, you have IB and IO. IB and IO. Again, B is dominant over O, which is why you get B blood group. So if at all you talk about phenotypes, that is physical nature, how many physical blood groups can you find? One is A, one is AB, and one is B. You do not count it twice, you count it only once. So basically 3 phenotypes are possible. 3 phenotypes, I have 2 options with 3 phenotypes, right? 3 phenotypes, I have 2 options. I will cut down the other options. Okay, what about genotypes? How many genotypes do you have? IAIA1, IAIO2, IAIB3 and IBIO4. So basically I have 4 genotypes. So your answer for this question is going to be B. So how many of you gave me the answer B? Ganesh, Preetham took away the answer. Okay, Preetham gave me the answer also. And Suma, Dhanush and Jessica and Kumada. Hello Kumada. Kumada also gave me the correct answer. So your answer for this question is going to be B for this particular reason. Is this clear, everyone? Can you give me a quick thumbs up if at all it's clear? And if at all, I can move ahead with the next question. Shall I? Shall I go ahead with the next question? Yes, thank you. Okay, 21st question. A pure breeding pea plant with the round and yellow seeds are crossed with pea plants having wrinkled and green seeds. Dihybrid cross. Okay, now on selfing of F1 hybrid of his cross, 64 progenies were obtained in F2 generation. Find out the number of F2 progenies showing non-parental character. Now this is something that we discussed in one short video of master stroke. If at all you heard me or basically saw that video, this question will be like this for you. Let me see who is able to answer this question without any help from Google, okay? Then only I will be considering it. So be very honest with yourself and try to find the answer. So you are basically crossing round and yellow and wrinkled and green seeds. And you are saying that at the end, at the end of F1 and F2, at the end of F2, basically you are getting 64 progenies. And out of 64 progenies, how many of them are going to show non-parental character? That means something that is not there in the parents. They are neither round and yellow, they are neither wrinkled and green. So Ganesh says the answer is A. Okay. Suma says the answer for this question is also A. Okay. A means 24. A means 24. Dhanush also says it is A. So everyone, try it by yourself is what I believe. Yes. GHM Gunashekar also says it, hello dear, and he also says it is A. Now listen to me carefully. For those of you who are not able to crack this question, it's very simple. We know 9:3:3:1 ratio. I spoke about it, right? 9 of them are going to be what? The dominant character, which was round and yellow. One of them is going to be the recessive character, that is what? Wrinkled and green. Now, this is what the non-parental character, that is, do not see in the parents. So over here, round was with yellow, let's make round with green now. Here also, wrinkled was with green, let's make it with yellow now. So wrinkled and yellow. So my dear children, if at all you calculate the whole of it, 9 plus 3 plus 3 plus 1 is equals to 16 progenies you generally get. You get 16 progenies. Out of 16, 6 of them are non-parental. Out of 16, 6 of them are non-parental. And now you are being asked about 64 progenies. Out of 64, how many of them are going to be non-parental? So 16 into 4 is equals to 64. 6 into 4 is equals to what? 24. So out of 64, 24 of them are going to be like this. Rest of them can be like this and this. Got it? So basically, your answer for this question is going to be A. Exactly, Ganesh Patil. Awesome, awesome. Wrote the entire thing also. Preetham also gave me the correct answer, dear. Okay. So for those of you who didn't understand, I hope you have understood it now. Similarly, you might be asked about this or you might be asked about how many of them are round and yellow. So please know about that. Okay. And it will be all sorted. So how do you calculate round and yellow? 9 divided by 16 into 64. So you will get the answer for that also. Got it? Give me a thumbs up if it's all clear. I'm waiting for your thumbs up. Then we will move ahead with the 22nd question. So this is from Principles of Inheritance and Variation. So we are having like one this, one that, and we are going ahead. One molecular, one Principles of Inheritance. Okay, thank you, Ganesh. Fine. Now, the next question that we have is question number 22. A series of experiments were conducted by Frederick Griffith in 1928 and transforming principle with which of this organism out there? So basically, Frederick Griffith did experiment on the mouse. Remember my Puchu mouse out there, right? So which organism was chosen for this particular experiment? That is a question. Thank you, Gunashekar. Thank you, Baswaraj. And Baswaraj, hello, dear. And also, thank you, Jessica. Thank you, Suma. Okay, thank you, Dhanush. Okay, guys, we are here with a new question. Let's try to solve this one now. Jessica says C is the answer. Nagaraj is there. Hello, dear. 22nd C. Okay, Preetham also C. Jessica also C. What about others? Dhanush says C. Okay. Suma says C. Everyone is in full josh. Oh my God. Streptococcus pneumoniae. Exactly. This was the organism that we used. And we had two things, R strain and S strain. Right? So yes, your answer for this question is going to be C itself. You guys are absolutely correct. Now, going ahead with the next one that we are having, and that is question number 23. Now this one again, everyone who have read through the NCERT or at least watched my video would be knowing the answer for this question. Experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in chromosome was performed on which plant? So basically, simple way I'll ask you this question. Okay, so you know semi-conservative mode of replication was tried to be proven by Michelson and Stahl experiment. Similarly, one more group of guys also came in to do some experiments on this, and for that, instead of using the different isotopes of nitrogen as used in Michelson and Stahl experiment, they used radioactive thymidine. So you are being asked on which plant did they perform this thing? That is the question. So Jessica says the answer for this question is A. Nagaraj also says it's A. Preetham also said it's A. Okay. What about others? What do you think is the answer for this question? This is so basically the people who did it, their name was Taylor and the colleagues. Okay, so Taylor and his colleagues, they did one more experiment to prove semi-conservative mode of replication using radioactive thymidine, and this was done on what plant? Just give me a quick second, dear. Hello? Nan live edukkuva, nan vilikam. Okay? Bye. Good evening, Shankar. Good evening. Yes, exactly. So it was Vishya Faba. Your answer for this question is going to be A. Fine. So everyone who didn't know it, you know it now. Good evening, Shankar. So this is how Vishya Faba looks, by the way. And next question is question number 24. In Morgan's experiment with Drosophila, when yellow-bodied, white-eyed female was crossed with brown-bodied, red-eyed male, and their F1 progeny were intercrossed, what was the percentage of recombinants in F2 generation? Oh my God. This will shake up your brain out there, right? Because it does shake up mine. So basically, this question is not as complicated as it looks. So listen to me carefully. Morgan did his experiment on Drosophila where he chose multiple characters all at once. Okay, so I'll tell you one very important thing. Over here, this is the gene for yellow body, suppose. This is the gene for white eye. Okay, these two genes are very close to each other. Now what do we have? Yes, that's all. Okay, so these two genes are very close to each other, you know that. Now, what was the percentage of recombinants in F2 generation? That is the question. So over here you had yellow-bodied, white-eyed female and also brown-bodied and red-eye females or red-eyed males. Anyway, when you crossed it, how much recombination did you see? That is the question. And you are giving me the answer as C, that is 1.3. Is that correct? 1.3 means very less amount of recombination. Is that correct? Yes. Why is it correct? Because these two genes are very close to each other. So suppose I am cutting this part and I am transferring it, both of them here. So what is going to happen? Nothing. It is just going to be exchanged over here. Yellow body as well as white eye is still going to be remaining together only. Similarly, brown body and red eye will also be together. Only that they are going to get swapped. So there is no chance of new recombinants being formed over there. There is only less recombination because if we talk about the lower region, yes, in that case you have recombination but very less. So 1.3% is very less recombination. On the other hand, I have a question for you guys and all of you have to try from your end. Okay, all of you have to try from your end. This is very important. White eye and miniature wing. White eye and miniature wing, how much amount of recombination do they show? These genes which are white eye and miniature wing, how much amount of recombination do they show? This is a very important question, again related to your NCERT. So when you say yellow body and white eye, yes, these genes are very close to each other, which is why recombination is less. What if the genes are very far from each other? Is recombination going to be more or less? Is the recombination going to be more or less? Whereas Madiha Khanum, yes, my dear child, your answer is C. And everyone, Nagraj also gave me the correct answer. And Jessica also gave me the correct answer. So 37.2, yes, exactly, it was 37.2. Here you do not have the options. Here you do not have that option. Yeah, you have the last one. That was 37.2 because they are very far up. Listen to me. The maximum amount of recombination that any organism can have is 50%, not more than that. And basically when these genes are very far from each other, now if at all you interchange, what is going to happen? The lower part can go there. The lower part can go there, which is why we say that yes, there is high chances of more different non-parental characteristics to be seen. So in that case, it will be 37.2, but here in this particular case, your answer is going to be C. Is this concept clear? This is important concept. This you should know. If at all it's clear, can you just give me a thumbs up? Yes, 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 Nagaraj and Madiha and Jessica. Dear Sri Raksha, I hope it's clear, dear, now. So 62.8 is never possible because 50% will be the highest recombination that is going to be there. Okay, so suppose now here and here it gets swapped. What is going to happen? These two guys are going to remain together, but this is going to go there, which is why we say that if you see in this terms, the new parental characters are going to be same. You do not get the older one that you had before. Okay, got it? Thank you, thank you. Fine. So yes, fine. Thanks, guys. 25th question. E. coli fully labeled with N15 is allowed to grow in N14 medium. The two strands of DNA molecule of the first generation bacteria have what? Give me the answer for this. Okay, thank you. That's so sweet. Yes. E. coli fully labeled with N15. it was fully labeled with N15. That means the DNA, both of the DNA strands were made of nitrogen compound which had isotope N15. Now you have been asked that they are now grown in N14 medium. What is going to happen? What is going to happen? Well, if the DNA is like this, of both the N15 strands are there, then they are going to settle down somewhere over here because it is very heavy. It is going to be heavy. So they settle down, very down. Now what about if at all they are grown in N14? Okay, so I see, ma'am, 15 days left for board. It's Habiba. 15 days are left for votes, but you have 15 days, right? And these 15 days are more than enough for you to give your best. So keep on trying, you will be able to ace it up. Okay, Dhanush says the answer for this question is C. Sri Daksha says it is C. And Preetham says it is C. And Suma also says it is C. Gunashekar also says it is C. Nagaraj says it is D. Is it? Is it D? Let me see. So, this was the parental strand that we had. The parental strand, parent strand, my dear children, had a heavy thing and it looked like this. Now, N14 media, if at all you are growing, listen to me. These two strands are going to separate. So basically I get a strand over here, I get a strand over here. What are these strands? N15, N15 strand. Now the new strand which is going to be formed is going to be N14. So this is N14 and this is also N14. So if at all you see, both the strands are different. They are hybrid strands. Why do I call it hybrid strands? Because one is N14, one is N15. Now if I try to centrifuge it, it won't go at the rock bottom. No, it will be somewhere in the middle. Why? Because it is an hybrid strand. So now the question is, do they have the same density? No, Baba, they do not have the same density. So this is wrong. Same density. Do they have different density? Yes, they have. But do not resemble with the parent DNA. So these two strands, do they resemble like the parent DNA? No, Baba, they do not. Which is why your answer is going to be C. Different density, but resemble with the parent DNA. Do you think they resemble? No, Baba. So which is why your only answer for this question is going to be C. For those of you who had said D, just check once. Nagaraj, dear, is it clear? Is it sorted? Or do we still have doubts? Okay? Guys, where did I keep the pen? Oh my God. So, was this the pen that I was using? Okay. Your teacher is a nutcase. That is me. Okay, I'm talking about myself. Okay. Let's talk about the next question. That is question number 26. Guys, come on. Okay. Gunashekar also gave me thumbs up. And with that, we go to the next question, which goes like this. Someone is giving me hearts. Thank you. For you also, so many hearts. Okay, from the chromosomal complements given below, identify the one which shows female heterogamety. That means females are going to produce two types of gametes. In which one of them do you see female heterogamety? In which one of them do you see female heterogamety over here? Okay, Jessica says D, Sri Raksha also says D. What about others? Preetham also says D. D is what? ZZZW. Where do you see it? Where do you see it? ZZZW. Ma'am, it's semi-conservative method of replication. Exactly, Nagaraj. Yes. So, where do you see D? Listen to me. What is heterogamety? As in two types of gametes are being formed. So usually in the case of human beings, we have something like this. Males produce one gamete having X chromosome, another gamete having Y chromosome. But females only produce one type of gametes which are known as homogamety, right? But here in this case, okay, here in this case, what is going to happen? There is ZZZW. ZW is actually female and ZZ is actually male. So males here produce same type of gametes, but females produce two different type of gametes, which is why we say female heterogamety. We say female heterogamety. This is seen in the case of birds. Okay, this is usually seen in the case of, yes, Madiha, it's seen in the case of birds, according to your NCERT, in the birds. Yes. Cool. So your answer for this question is going to be D. Now, how many questions are left? Only 4 questions are left, and after that, we are going to get independence. Okay? At least you will get independence. Okay. And yes. So, question number 27 is Sidhu, it is second PUC. Okay. Both male and female have normal vision, though their fathers were colorblind. Now this is a tricky question, everyone pay attention, okay? Both male and female have normal vision, okay, though their fathers were colorblind. Their father were colorblind, okay, and mother did not have any gene for colorblindness. The probability of their daughter becoming colorblind is what? So listen to me carefully. When I say that male and female have normal vision, with that I understand that male definitely is like this, but females, my dear children, can definitely be a carrier. Why do I say that she can be a carrier? Because here it is clearly written that the fathers were colorblind. Fathers were colorblind means it was something like this, and this X chromosome goes to the female only, not to the males, right? So over here it might have went to the female. She might be a carrier. Carriers do not have any vision problem. They are just normal people. Normal vision will be there. They will be just carriers. Now over here, okay, and the mothers do not have any gene. The probability of their daughter becoming colorblind. Now if at all I talk about them, what is the probability of their daughter becoming colorblind, my dear children? For a daughter to be colorblind, both of their genes should be coming together, right? So from here also you should colorblindness, here from here also you should get colorblindness, but there is no male. The male is not affected at all. So will you get anyone who is going to be colorblind? She will be only a carrier, cannot be colorblind, because you need this chromosome and you do not have that, which is why 0%. So I saw a lot of people saying A, that is Jessica, Madiha, Dhanush, Nagaraj, and Suma, and Preetham, and Sri Raksha. Awesome! Oh my God, do you know that you guys are awesome? So initially when I started off this video, I was like bit sad seeing, okay, probably no one might have watched the masterstroke conceptual videos, which is why there are wrong answers being given. But now I see that you guys are giving me all the correct answers. Just give me a second. Okay. So this is clear. Shall I go to the next question then? If this is super clear, shall I go to the next question then? Okay. Yes. Question number 28. In which region of the tRNA molecule is the amino acid binding site located? In which region of the tRNA molecule is the amino acid binding site located? Ding, 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 ding. So my dear children, tRNA is a normal RNA having 5' end and 3' end. You are being asked on which end does it attach to amino acid? That is the question. Hello Shavina. A very big hi dear. C. Okay, Masha Ali Khan says C. Okay, hello dear. And Jessica also says C. Dhanush says C. Nagra says C. Shabina Taj also says C. C is what? 3' end. Yes. So this is the amino acid acceptor end. Here only amino acid is going to attach. And always remember that here, this is process known as amino acylation or charging of tRNA, and it requires high amount of ATP. It requires high amount of ATP. Okay. Madiha Khanum also says C. Shabina Taj also says C. Suma also says C. Yes, my dear children, it is C. By the way, everyone who is preparing for KCET, now I can go to the slide that we just missed because we are anyway gonna end the live. Any person who is preparing for KCET, and if at all you want to try one mock test for free, totally for free, you can go ahead and go to the link given below in the description section. I hope it's there in this video also. If at all it's not there, you can just go to see the master stroke of evolution chapter. It will be there below over here. Enroll for this free KCET PCMB mock test PDF. There's only one PDF. Okay. What you have to do is click on that link, put in your mobile number, and this particular PDF will be ready to download. Download it and try to solve the question by yourself. Okay. Got it? So please try to go ahead with that if at all you want the KCET mock test. And remember, it's a free thing, so definitely you can try it off. So go try it up now itself. And everyone who's preparing for KCET, we had already launched the ABHYAS KCET test series. I hope you guys know about it. There are 35 mock tests. Do not do the mock test now. Go ahead with the chapter-wise PYQs, the one that we are doing right now, okay? So I have handpicked a few questions out there, but you can go ahead solve more questions because there are definitely more questions. And all of it will be under proper timed condition, and the pricing for this is rupees 1499. It's remaining the same only like the previous year. We just added one more thing, and that is chapter-wise PYQs for you guys. And because we saw that lots of children got the benefit of it the previous year. So out of 3 lakh children who gave the examination, almost we got ranks like 171, 443 and 693, and the list goes on. Almost thousands of students got into the best colleges out there. So this is not something that you should be missing out on, but please do your research and then only go ahead. Again, the link will be given in the description session. Okay, so please check out both of them. And now we will go out to this particular question, which is, yes. So this is a 3-end. Okay. 29th question, this is going to be the second last question, my dear children. Choose the possible genotypes responsible for the lightest skin color in human beings. Lightest skin color. Ma'am, bio important MCQs for board exam. Shabina will be coming up soon with marathons. There will definitely let you know what are the important MCQs. We will solve it with you guys. Okay, and thank you for coming up and telling that. And Gunashankar, you said thank you. You're most welcome, dear. And ma'am, your voice is so pleasant and the way you encourage is like energy booster. Thank you so much, Jessica. That's so sweet of you as well. And you guys also so sweet, even though I'm not able to hear your voice, but I do definitely see your comments. And when I see some very sweet comments are there, it instantly makes me happy, and also I feel encouraged. Yes. So children gave me the correct answer. Sri Raksha says C, Jessica says C, and Preetham says C, and yeah. So C is what? A, B, B, C, C. Why do you say that someone who is having something like this will be having the lightest skin color? Because of one very big reason. Listen to me. This is an example of polygeny. Again, something opposite to what Mendel was saying. Mendel said one gene, one character. But here it is not like that. Multiple gene, for example, capital A, capital A, capital B, capital C, capital C. All of them, when they show up together, it is something like this. And basically that shows the darkest skin color. All of them are able to express themselves, which is why darkest skin color. Okay, multiple genes are affecting over here. This is an example of polygyny. Now suppose I add up one recessive allele over here. A, B and C. So it will be capital A small b, sorry, capital A small a, capital B small b and capital C small c. This will be lighter than this. Suppose all of them are small ones. What is going to happen? You will be seeing the lightest skin tone, that is small a, small b, small b, small b, small c. This is the lightest skin tone. This is applicable for two things, that is your skin color, and also one very important thing, that is what? Height. Okay, that is height. So skin color and height is something that is there. Yes, recessive condition, exactly. So your answer for this question is C. And now finally we move on to the 30th question, which is the last question. In a crime investigation, the investigating officer collects different biological samples from the crime spot for DNA fingerprinting analysis. Which of the following samples is not useful in this analysis? What do you think? So thank you for giving me so many hearts. Someone is giving me constantly so many hearts. So yes. Guys, so which of them, for example, if at all you want to do DNA fingerprinting, which one is not going to be helpful? Skin shreds, erythrocytes, semen sample, hair follicle. Which one is not going to be helpful? The class, this class means a lot, ma'am, so thank you for this. Oh, that's so sweet of you, Sri Raksha. Thank you so much for telling that. Okay, Jessica says B, Chandu also says B, and Ganashekar says B, Madiha Khanum says B, Sri Raksha also says B is the answer. Why is B the answer? Why erythrocyte is the answer? You definitely take blood, right? Blood is also a place from where you take samples. But why is B the answer? Because erythrocytes, my dear children, does not have nucleus. That means no DNA. Erythrocytes, RBC, does not have nucleus. That means no DNA. So yes, you take blood sample, but in blood you have multiple other cells also. It's not just RBC, right? Yes, Jessica, does not, because erythrocytes does not have nucleus. So that is a reason. So I hope you got the answer for this. Why? Because they have to carry lots of hemoglobin. That is why they do not have much space to have the DNA. Ma'am, in the last few days before boards, what should I focus on? I'm planning to solve 10 years previous year paper. I have already revised. Should I solve any annual papers or supplementary? Solving 10-year previous year paper would be more than enough if at all you are someone who studied everything. But I will always request you to please go ahead with question bank questions also. So in the question bank also you find the previous year questions, right? So I'll recommend you refer to the question banks. You will find newer questions and the older questions both. Okay? So please do that. So your answer for this question was B. And before I end this live, I would still love to talk about the videos that is already done. Children, if at all you're not taking it seriously, I'm telling you again. So we started off master stroke conceptual video in a hope that everyone who is struggling with concepts as of now, it will be helpful for them. Okay, so I have done videos until the chapter. I think the latest chapter that I did today was, just a second. Where is it? It was microbes in human welfare. Okay, so microbes in human welfare, until that particular time, it's done. Go make sure you watch the videos. It is super helpful for your PU2 as well as for your KCET. It is definitely going to help you out. Watch the conceptual videos, share it with your friends, and yes, that's all. That's the only thing that I have to tell you. And with that, I end up this particular live. So Preetham, ma'am, will you do KCET after boards? It will be great if you do. Yes, we'll surely do that after boards, is what I believe as of now. So yes, the team has to decide on it, but yeah, we are totally aligned with that. Okay, Preetham. Gunashankar, thank you, dear. Thank you. So everyone who joined today's, ma'am, do human health and diseases. Human health and diseases chapter that I have done. Okay, I have done the chapter, but yes, I have to do the MCQs also. In the next live, I'll be dealing with 2 chapters, human health and diseases and microbes in human welfare. Okay, and evolution is also there. Yes. Love from Bellary. Oh, that's so sweet. Love from Bangalore. That is from me. Chandu, thank you. Okay. And yes, human health and disease, if at all you're looking for concepts, I have done it. Okay, conceptual video is done. Chandu. So everyone is from Bellary. Chandu is also from Bellary. Thank you so much. And children, before leaving, make sure you do not forget to click on the like button. And if at all you're a new person out there, do not forget to click on the subscribe button as well. Ma'am, please make a motivational video before the exam. So there is a video that I had made today itself. I will be forwarding that to you. I think it's there on the YouTube channel now. But yes, I can definitely share it with you as of now itself, if at all you want. So here it goes. Please make sure you watch this video. Listen, I'm not a motivational speaker, but I definitely try my best to keep you guys happy, okay, in the ways that is possible by me. So there is a video that you can check it out, and please feel motivated. And thank you so much. So I'll end up this live. We will meet really, really soon with the next live again. And lots of love. Bye-bye. Please take care. Thank you, Madiha. And yes, I got it, Preetham. Definitely, I'll be doing that. Okay. So children, do not forget to hit the like button and subscribe button. And watch the video if at all you feel that you are demotivated. Yes. So, yes. Thank you, Suma. Bye-bye. Take care. Thank you, Rishi. I hope the videos helped you well. Well, hi, I'm seeing your comment right there. Okay, bye-bye. Take care.